Okay, well, let's start. And I think the rest of the people will join. So, so there is this morning workshop that is dedicated to the omics analysis of skeletal muscle under normal and pathological conditions. So you will see three talks. One from Gisèle Bonne about the European level. A second one that will be, so I don't know, it's probably not non-pathological condition, but you will see that Fabrice Bertile will discuss about some specific conditions impacting or not the muscle. And then Alain Meyer, after the break at 10:45, will discuss about the inflammatory myopathy. So this workshop was organized by the European Consortium CoMPaSS. And now I would like to welcome Gisèle Bonne, who will tell us about everything she wants to tell about Omics and Europe. Thank you.
Thanks a lot, Jocelyn, for the introduction, and thanks for the CoMPaSS Consortium for the kind invitation to this workshop. It's really my pleasure to be there today. And before to start, I just want to say that what I will present, it's a summary of previous EU projects and ongoing EU project. And I have some slides that I borrow from both Holm Gressner, who was the leader of the previous SOLVE-RD project, and also have a few slides borrowed from Daria Julskowska, who is the main coordinator of RDERA project. And so now I can enter into the topic. So those two projects, I will focus on omics analysis, focus on the trying to solve unsolved cases in rare diseases, and of course, with a focus on rare neuromuscular disease. That is the focus of our discussion today. So, SOLVE-RD, as I just said, was an EU project funded in 2018 for a bit more than 5 years, and was gathering 13 countries with 22 beneficiaries. And among, on top of the partners participating to this project, there was some rare disease expertise and infrastructure on board, like ERN, European Reference Network, RD Connect platform, EGA, European Genome Archive, Orphanet, Urgentest, and Eurordis for the patient representative. So mastering all, as much as possible, all the stakeholders in the field of rare diseases. So it was well spread around Europe, with more than 20 different groups contributing, more than 300 clinical scientists involved. And as I said, at first there was 4 ERN, Ithaca, R&D, EuroNMD, and Genturis. So Ithaca is for developmental defects, newborn. R&D is rare neurological disease. Neuromuscular disease, and Genturis is rare cancers. And later on, 2 additional ERN joined with Epicare for rare epilepsy, and Rita, I missed the topic, sorry. I should know, but I missed from now. And there was also some undiagnosed rare disease patient program. I think it was in Spain that has been involved. So the concept was to reanalyze finding causative genes in patients without a diagnosis, meaning that more than 19,000 datasets were reanalyzed. And also there was new omics analysis set during the project for unsolved cases. And on top of this reanalysis of data, and I will enter into the detail in a minute, there was also some money, part of the budget, for sending grants to just establish some functional analysis for new candidate gene to make functional analysis with a network that has been established to do some matchmaking between clinicians having a candidate gene and basic scientists working on a model, on a system, on a pathway, far away from the rare disease field, but where the genes were involved. So putting together these basic scientists with the clinician, with their data, just to establish model and make functional analysis. So for the reanalysis part, the project gather data from the different ERNs I just mentioned, and they're organized in different type of cohort. The main cohort was cohort 1 with unsolved cases with more than 19,000 unsolved exomes or genomes that were previously analyzed locally, but without any results. There were also some cohorts that was assembled within each ERN, like for neuromuscular disease, there was some myofibrillar myopathy cohort, there were some congenital myopathy cohort, or just assembled among the partners within the ERN, unsolved cases to make sense and analyzing all together the different cases and to compare them together. There was also a cohort called ultra-rare diseases for very exceptional cases. And again, you may have one case in Portugal, another case in Lithuania, another case in UK, and put together and analyzing at the same time these cases would make sense and perhaps find some common variant in some genes. And there were also a cohort, a few cases called unsolvable that has been explored by everything yet until the start of the program. And for those, there was additional omic proposed like metabolomics, proteomics, and RNA-seq, genome, and just putting all together this data to try to solve those. So that was at the beginning of the project. So what there has been several publications now on the concept and how and what has been achieved and how it was working. So the concept was to share again all the data and to have them reanalyzed just with the objective to have the possibility to solve additional cases. And so how it was organized. So first of all, I was touching a bit on this yesterday during our discussion, is that first of all, the patient has to consent to share the data abroad most of the time, because the data were collected through the RD Connect infrastructure, EU infrastructure, and also called GPAP for Genome Phenome Analysis Platform, which is located at CINAG in Barcelona. So apart from Spanish patient, every other patient needs to reconsent for share the data outside of their country. And so then the data was submitted on this platform with genomic raw data or exomic raw data, the phenotypic data coded by HPO term and ORFA code, and also the detail of how the genome or exome have been performed for the metadata analysis. So there was a standard protocol setup And the raw data were analyzed again, new alignment, no new call variants, and together put in front of the phenotypic and the pedigree, and all the collected data was stored at EGA. And so every partner could have some teams to analyze data and that can connect to the system to have the local cluster to reanalyze the data. And there was so a space in the cloud to make those analyses. And all the infrastructure was built during the project. So how it has worked is that there was two levels. On one side, among the partners, there was bioinformatician, a computing person, geneticist who developed algorithms and filter tools to sort out and reanalyze the variant. That was so-called data analyzing task force, gathering people from the different partners. And they establish tool and they analyze the data. And they make reanalysis of all submitted data as batches. So analyzing 500 or 400 cases at the very same time. And on the other side, so the output of this was some list of variants. So we received list of small indels variant filtered among all the cohorts submitted. We had received copy variant number variant. We have received structural variant. We have received de novo variant. And each time you have to look, this data was sent to the other group of experts, the data interpretation task force that gather clinicians, again, geneticists, and that try to make sense about the list of variants they received in front of the phenotype and the clinical presentation of their case they have submitted. And the discussion between those two end up with some cases that has been eventually solved. So I will jump on the final result of this project that has been published recently in Nature Medicine. And it's all the final number of the Solverdi project. So as I just explained, there was this submission of rare disease data, a comprehensive reanalysis, and there's two expert level review of the data. So on one side, all this number of genomic data sets submitted with a mean average of 6 HPO term, which sometimes is just too low and better described as a clinical phenotype, better it is. And also the pedigree information to make sense of all this together. So through the pipeline I just tell you, there was all this reanalysis of disease gene. And what I did not tell yet is that the reanalysis was performed not on all the existing gene on the genome, would have been too much work, but it has been done for each ERN on the known disease genes. And for neuromuscular disease, it was analyzed against the list published in the muscle gene table that we update every year and that is published in neuromuscular disorders. So these are the numbers for RO-NMD, and that you can see that we end up with solving 6.3% of cases among all the data submitted. And you can compare with other ERN, and you can have the feeling that it was not highly performing. But eventually, we need to keep in mind that the data that have been submitted to this reanalysis have been before analyzed extensively in each local reference center. And that was really the unsolved cases that were submitted. And so those who were solved were either variant in big genes that were not well covered locally, or new recently published genes that have not been evaluated locally before submission, or, and it was most of the case, some structural variant, copy variant number. All these variants that are very difficult to detect easily with short read sequencing and classical analysis tools at the time of the beginning of the project, because we started in 2018 and it's ended up in 2023. So keep in mind that as the pipeline and the algorithm we use to filter data go fast and evolve quite fast, we need to keep this in perspective. So just to highlight one of those difficult cases we had, this was a family that has been explored in France for more than 20 years. I think the family traveled to one reference center to another reference center. They were living in Bordeaux and they moved to Paris and tried to figure out what they had. And so it was to the family. We have the DNA of the people with a circle that has been exome sequenced. And it was to the diagnosis at first was congenital myopathies, but nothing came out on the classical analysis. And with the classical pipeline we had at that time, the WES data, the exome data was highlighting potential candidate genes, but nothing very exciting and nothing that was making real sense at that time. And we were a bit going in circles, so to say, and not very keen to push forward a lot of functional analysis without additional insight to go over. So among I don't remember which was the first round on reanalysis of the second, but at some point, German Demidov in Germany contact us and send structural variant feedback. And there was 2 lines. The Excel sheet was quite long with a lot of variants. So we saw them one by one. And for this one, there was a structural variation in titin. And at first, we were used to have multiple structural variants in titin. And we answered false positive, full point. And then he sent back us the file and said, it's not possible. This one, I'm pretty convinced that it's a true one. And to push us behind our thought, he just sent us this screenshot and said, you see, I'm sure there is something, because the depth of the reads was quite different. But of course, when you look at your exome data, you don't go looking at the read coverage on every genes one by one. So the tool he used was able to detect this big deletion in titin. We further explore it in the family. We check the segregation. We make RNA-seq and eventually find that there was in frame large deletion in the titin in this family that was well segregating. And we, of course, we've been discussing with Marco and Mireille Cosset in France, expert in titin. And eventually we put together a series of patients with in-frame deletion. So this was the one we had in titin for our family. And it's eventually get a nice paper together with our collaborator within Europe on all this rare in-frame deletion in titin, and I will not go into much detail in front of our biggest expert of titin here, I mean Marco. So, as I say, the Solverdi program was quite efficient by the concept of having this harmonized reanalysis of all this data set together. There was, as I said, a substantial portion of novel diagnosis that was different type, that was not a small indels or point mutation variant. And the organization by data analysis task force, followed by data interpretation task force, occurred to be quite efficient in a reanalysis of this large amount of data set. And the coordination team makes some calculation that it was less than 30-minute expert time per patient. Personally, I spend a bit more time than 30 minutes. I still don't understand how they came along with this timing, but more statistical calculation than true time. So now I will move to what I used to say, it's SolveRD 2.0, the following project. But the following project of SolveRD is embedded in a much larger project, which is called ERDERA for European Rare Disease Research Alliance, that has started last September and which will last at least for 7 years. This is a EU partnership, meaning a partnership between member states and the European Commission. And I will come on that in a minute. And with EU project, for this type of partnership, you need to have big objectives. And there is 3 general objectives for this project. The first one is to speed up the diagnosis of unsolved cases, again, and with diagnostic established or enrolled in systematic research in an average within 6 months after coming to medical attention. That means for every citizen in Europe having access to some expert within 6 months, wherever they live in Europe. The second main objective is to speed up treatment for rare disease. So new effective therapy for rare disease approved, with a majority of which addressing disease without approved option. And the third one is to push forward a better understanding of the impact of rare disease on patient, family, society to improve their quality of life. So that has very general objective for a lot of partners for more than, let's say, 7 to 10 years coming. So the project gathered 181 organizations from 33 countries for a global budget of 380 million, among which 150 million is provided by the European Commission. The rest is provided by the member states. So some of you may have already seen these beautiful houses and, you know, EU projects like work packages, etc., pillar. So I will try to explain the main points, the main work package, the main aspect of the project where you can get access, benefit for it, or contribute for it, and just to highlight a few points where it could be an added value for rare disease expert, clinician, researcher working around. So this house is based on a funding work basis, which is the national capacity, which is in every big EU project. The former project on rare disease was called European Joint Project for Rare Disease. And ERDERA is a follow-up of this, extending the concept. So for this project, there is national mirror groups. That is, at the national level, the rare disease community make connection with what occur at the national level and connect with European level. Then you have the wall of the house, and I will enter into the detail of the clinical research network, the different type of funding you can have access to. There is some infrastructure that are continuing to be built by different teams contributing to the ecosystem. And on the top, you have the coordination and the strategy at the European level, but also mirroring what occurs at the national level. The objective is to go all together in the same direction for gaining results for the main objective I just highlighted. So I will briefly go through this different part of the house, starting from the national mirror group. Just, of course, you say nothing to do with omics and the talk today, but it's just to highlight when you back in your respective country, you may be an actor of this national mirror group, as what occurs in your country, whether there is network organized or a rare disease plan or whatever, you can network with your national counterpart and try to make some lobbying to discuss with local authority just to align what occurs at the national level with what occurs at the European level. The objective is that what occurs in different member states is not orthogonal to what we try to do at European level, just to be in the same direction, going to the same, trying to push towards the same objective, the three main objectives I highlighted. So a few words on the different funding opportunity, and I know that for researcher, it's always very important to get additional money for ongoing projects. So I'm sure that most of you know the Joint Transnational Call. They have been established a long time ago. The concept is still the same. It's every member state puts some money through the national research agency together with some EU money as well. And every year there will be a call. There was one that just went over a week ago. And so a new topic will be released every year. Usually by November, December, it will be open with submission by end of February or beginning of March. So the main goal is to enable scientists in the different countries to build effective collaboration on different topics that will be highlighted. And so usually it's the GTC are two-stage process, letter of intent, and then if you pass, you can go through the full program. It's a 3-year project, and you need to have 3 to 4 eligible partners with a maximum of 6 per project. And importantly now, it's almost mandatory to have patient representative as partner, and they will receive as well some money, which was not always the case before. It's just to emphasize that in ERDERA, the patients and the position of the patients are very important. So they must be part of the project. They must interact. There will be also some networking support scheme that was also existing in previous EGPRD project. You can get up to 30,000 euros per meeting or network organization you want to set up. You can apply continuously. I'm not sure if it's open yet, as the project started only last September. And you need it to be very collaborative with a minimum of 3 applicants from 3 different participating countries. And what is new within ADERA is that there will be some funding for clinical trial. It's not yet open. The first call might arrive in 26. And for this, the European Commission put 30 million on top of the budget we have negotiated at first. And there is still discussion about how will participate the patient, but it's important that they will be there also as well. So, as I said, you have the funding, you have some support service infrastructure. Just to highlight again, because it can be helpful for people working in the field. And sometimes we don't know all what is already existing at the EU level as infrastructure, as support to help us to develop our project. So in collaboration with EATRIS, there's been during EGPRD some management tool that has been established. That is, when you enter the field in rare disease, if you have no clue about what exists as tools, as infrastructure, you can contact this address. And you have, whenever, you can be at different stage of your process, of your research, of your career, of your knowledge about rare disease. And they will help you to find good experts to help you, just not to reinvent the wheel, but just speed up your development of your rare disease field. And so there is more than 400 different resources and use case that you can look at the different websites highlighted here. And there is a lot of different type of expertise. And I can highlight, for example, which is an expertise that I'm always missing, that it's really a burden for me, which is the regular and the regulatory aspect, the ethical aspect, which is for a researcher really a burden on the daily, in my daily life, I would say. So, and you can see that there is a lot of different topics and resources already existing. So it might not be your favorite disease, but you may have some similarity, and you can find some very interesting tools there. And there's been a lot of work achieved already by others. And again, the objective is not to reinvent the wheel, but just to find the experts that can help you to go faster. The same line, there is some clinical trial toolbox and methodological support. This has been developed in collaboration with ECRA, another EU infrastructure. And again, look at this web address. There is a lot of training for those who go from preclinical to clinical trial readiness. There is different teaching from different type of people, and it's just where you can find all this. Also, I will speed up because I see that the timing is running. There is a virtual platform that has been established during EGPRD. It's all what they have assembled together. Again, a lot of resource and pathway, etc. And this will be further extended within a so-called rare disease data hub as part of ADERA. And there is a lot of experts putting together tools to have interoperable registries and data sets. A lot of education also. Some MOOC have been developed and will be further developed, and there has been EuroNMD that has contributed to one of those. This will be, again, further expanded at all levels for training at any stage for research, for diagnostic research, for clinical research, for empowering the patient association, etc., just to have a complete and detailed ecosystem where everybody can find his way in the middle of this very complex situation of rare diseases. And I will end up quickly on what will be SolveRT 2.0. It's here, it's part of so-called one of the two walls of the house I highlighted as a beginning. It's a clinical research network. Is within ADRA, there is ongoing project, use case, and for diagnosis, there will be, what I used to say, SolveRT 2.0, that we will expand the concept developed using within SolveRT and extend it to not only 4 European reference networks, but to the 24 existing reference network. Instead of reanalyzing only the known disease gene, they will extend to larger set of disease gene. They will also look at long read. And so, as I just said, we will move from there to a larger diagnostic workflow, and I will detail this. And this will be based on the work established and the infrastructure formed during SolveRT. There will be also innovation developed as part of RDRA to extend the concept of SolveRT, but there will be some connection with other EU projects that are ongoing, such as the genomic data infrastructure, which is connected to the 1 million genome project, where there is team working to assemble the general population genome data set. And so they are working at assembling all the genome data of the general population in connection with UK Biobank, etc., etc. So they are dealing with a massive amount of data, and they're building a federated infrastructure to interrogate all this. So, and as part of this project, there is some use case that are rare disease. So there will be connection. Again, EU, what they want when they propose and they put money on the table, they want that the money they put in the different EU projects is going back to every European citizen, and they want that every EU project connect together. And for the rare disease, there is a lot of money around, and RDA will be joining other big infrastructure to make sense and again to speed up the whole process. So for rare disease diagnosis, we will have SolveRT organized in 3 main work package. One will be the coordination to get all the data in correct way, with all control for data access, quality of data, collection of data, either, as I said, in a distributed way, all upload on RD Connect GPAP platform in Barcelona, but in the future, staying in the country and access through a federated approaches. So this will be the management of the data. Then there will be within work package 7, all the reanalysis as a concept I explain you, making data analysis force, data interpretation force, developing new pipeline, extending the existing one, improving the existing one. And for underrepresented country, there will be possibility to make some new, to generate new genome data, exome data, or other omic data. The budget is dedicated to underrepresented country, those who may have more difficulty to have access to those new technologies that in other member states we have access to. So work packages will be aligning all the data, Then it will go through HCP, the Clinical Reference Center, will provide to ERN the data. They will be analyzed by the Data Interpreting Task Force that will release things, and the Data Interpretation Task Force will analyze this. So we will have what would be called super DIFT, assembling several ERN together. We will, for NMD, they will work with rare RND, rare neurological disease, because there is a lot of overlap for some clinical entity, and there will be two other main DIFT, super DIFT group. And as I said, at first, everything will be organized in a distributed way, built on the concept of Solverdi, uploading data in Barcelona on RD Connect. But progressively, as the infrastructure will be built, the data can stay in the respective country and be interrogated at distance. So all this will be organized with different partners. And so just to finish with, there will be, for those who are more on the clinical side, some work packages that will work to updating and pushing further the outcome research, the real world data and the clinical outcome assessment to improve the tools. And for this, it's nice title of different tasks, but behind this, there is different disease focus that are used as a use case to develop the concept and validate the new tools that could be further expanded to other diseases. And as you can see, NMD, neuromuscular disorders, are implicated in the different tasks of these different work packages. And finally, within the clinical research center, there will be also some development of innovative therapy and how to push forward a clinical trial with changing the concept of trial for rare disease, adapted to rare diseases. And again, it will be focused on a few diseases as use case. And once those new concept, new development will be validated, they can be adapted to other rare disease. And so beyond all this, as I said, there is multiple ongoing EU projects. And I realize that on this slide, we miss CoMPaSS. But you see that there is a lot of opportunity at EU level to work on rare disease, on omics, but not only, because rare analysis, unsolved cases is just the starting point. Once you solve your cases, you need to understand the disease mechanism. You need to develop your treatment, you to go to the clinical trial and back to the patient. So I hope that I have in part highlighted a bit what is behind this big project, and happy to answer any question, and feel free to contact the coordination team of RDERA for further development.
Thank you, Gisèle. So are there any questions? Marco, yeah.
How will really complete so much? So how we will reanalyze post-data, let's say? Is it something that we will do in a similar way compared to how we used to do? How we will look for new genes, for example?
What I understood is that the data that has been uploaded for Solverdi will be pushed further to RDERA, and they will extend the gene list. And what I understood, but I'm not part of the data analysis task force, is they will develop new pipeline or extend or reinforce the existing one you may be part of it, I don't know, and probably to extend the infrastructure to be to have more calcul power and space to be able to manage the interrogation of a larger list of
Yeah, but what I mean, so the point is, so the data will be constantly reanalyzed.
Yeah.
And that's why I think also in our project, it will be beneficial to have data on RD-Connect, because I guess the idea behind this is that data is collected now and they will be constantly
Yeah.
reanalyzed.
what I
almost
understood. Yeah.
in a semi-automatic way, let's
Mm.
like that.
And what I think, having heard all what you are developing in CoMPaSS, and I was discussing this with Elisabetta yesterday, I think the tool you develop, the platform for the phenotypic characterization, it's much far away from the 6 HPO term that were in an average uploaded. And you can see, I'm sure you did the work. When you find a variant, you contact someone who uploads the case, and they have put a few HPO terms. You say, well, it's fitting my own phenotype, and you contact and you realize that not at all, because there was very generic HPO term, and when you go into the detail, it's nothing to do. So again, the gene variant, it's something, but without all the clinic, the histopathology, the imaging, you cannot make sense of it. It's so powerful to help for the functional validation further. If you stick to only the variant and a few HPO terms.
Yeah, in fact, this is something that is very important, and I strongly believe in it, that this fine description of the patient, the more data, phenotypic data we put, it is much clearer to understand. Because what I found is that we are looking for new genes, but I think we have to, and this is what CoMPaSS want to do, is to change the perspective, starting from a different thinking of more elements that contribute. And because a variant in a certain genetic background can act in a way, and in another genetic background can act differently. And the phenotypic description is very important.
At first, for Solverdi, the aim was to, one of the work package was just to reassemble code upon merging HPO term or ORFA code. But I'm not sure it has been so efficient so far, because again, the clinical description put on GPAP was very minimum for most of the cases. Because when you upload 600 cases, it's hard to make all the detail. It's again the discussion we had yesterday, how much time you need to upload your data in a sensible way. You cannot expect big result, interesting result, if you don't tell what your patient have. And it's not saying, well, he has a neuromuscular disease. Okay, fine, but what type?
In the end, it's a wonderful project because it brings together a lot of elements the links we were missing.
Mm.
But on the other hand, what I see here is we need a lot of people to do that. And I think that's something that's completely impacted that perhaps really on all sides, research side, clinical side, and even our systems. So this will be the bottleneck, what I see here. But therefore, what I miss is the implementation of this all in the health system. It needs to come down really to our daily situation where you can implement it, start with that directly electronically.
And ideally,
some of the Scandinavian countries have that.
Yeah.
But
there is still, When I present this in France, I can tell you, all the physicians go away, entering again and again data. And I think the other project, which is Jardin, which is gathering ERN, which is more on the clinical side, not the research one, but the clinical and the healthcare system, is just to empower the local healthcare system to have all this electronic record with the code from scratch, from the out clinic. You enter everything, and all the dream is to have every healthcare system having the connection and code encoded data from the very beginning, and then everything will be interoperable using FAIR concepts, etc. And then you end up in research part. Already everything has been pushed in the pipeline automatically. So I remember when we discussed last week at EuroNMD General Assembly, it was all about AI helping this and extracting from clinical records. Okay, we can say it, but we have to do it. And that's the other point. But if I go backwards and think how it was in 2018 when we start Solverdi and now where we are, again, you can keep in mind the 6% of solved cases, which is not much. But we have built an infrastructure. We have learned how to work with that, how to, we train the clinician to do that. And it's a slow process, but I think everybody is moving in the same direction and eventually it will work. I feel that we are just impatient because we would like our respective cases that are working for years, trying to solve them, are still unsolved, but
yeah, that's why coming back to CoMPaSS here for our situation, we need to try to implement our data base. This can be interconnected with the healthcare systems. So therefore, I was always interested. All needs to be done on the cell, because that's the easiest part to connect. Otherwise, you're not going to be able.
And I think in CoMPaSS, you need to establish the platform with source cases, of strong background, and then training the system with a very strong data, and then it will help a lot. It might be a burden at the beginning, but once you did that, the training will be much easier.
And therefore, we connect with the HPO terms and the author.
Yeah, yeah. And I know that the OrphaNet teams are very keen to exchange and discuss, to adapt missing code, etc. So feel free to contact them at any time.
Okay, thanks a lot, Gisèle. So now we change gears and we will go to the analysis of a sleeping animal by Fabrice Bertile.
Do I stop the partage? Be able to launch the. Oui, c'est sur le bureau. Perfect, thank you. Good morning, everyone. And first, let me thank the consortium for this kind of invitation. I'm glad to be here and talk to you about today some of the results we obtained in hibernating bears and notably around muscle proteomics analysis. Hibernation, first, I would like to define this term. You know that it's an adaptive strategy that allows various animals to survive harsh environmental conditions, such as cold winters, reduced resource food availability. And physiologically, this state is characterized by torpor. And torpor, it's a situation where everything is done to save energy. So the animals are physically inactive, they reduce food intake. Most of the time, they reduce body temperature. Metabolic rates are also largely decreased, heart and breathing rates too. And finally, there is suppression of all the energetically expensive processes in these situations, such as protein synthesis, ion fluxes, or enzyme activities. Here is, for example, the annual cycle in hibernating brown bears. You can see here that those animals are hibernating during 5 to 7 months a year. So they only have 5 to 6 months to grow up again their lipid fat reserves that allows them to survive this period. And as you can see, for example, in this brown bear female from Alaska, here you can see that 2 months after emergence from the den, she's still a bit emaciated, but 2 months later, just before denning, she has accumulated a large amount, sometimes in adults, up to 70 to 100 kilograms of subcutaneous fat. In regions where berries, rather than compared to here, rather than salmon are available and the main constituent of the bear diet, they are ingesting around 20,000 calories a day during the summer to build up the lipid reserves. So what is interesting with hibernation, as previously said this morning, this is not a pathological situation. And it is striking to see that despite those classical atrophic conditions, physical inactivity, total fasting, and despite other metabolic features, the animals do not develop any pathology. Their different organs are preserved, their function are preserved or slowed down, and they come back to normal upon emergence. And they also do not exhibit osteoporosis or atherosclerosis or thromboembolism. There is a decrease in insulin sensitivity during winter, but everything is restored during summer. There is no heart failure. And what will be interesting for us today is that they do not exhibit a severe muscle loss despite these conditions. So this is our focus. And here you can see that during the first months of hibernation, they lose, for example, for this muscle, I chose here up to 15 to 20% of protein content. And this is one of the muscles that is the most affected. But this loss remains stable afterwards during the 3 following months. And it's the same for muscle strength, which is decreased by only 20% over 4 months of hibernation. Of course, in humans, the loss of muscle protein would be largely higher here. And it has been measured, for example, in inactive humans, it was a bed rest model, that they lost 54% of their muscle strength over 3 months of inactivity. So how is the protein balance regulated here? The situation is in humans, for example, in inactive humans, there is an imbalance that develops because synthesis are decreased, but most often muscle protein catabolism do not change. Contrary to this situation, in hibernating bears, the balance is maintained because both synthesis and degradation of proteins are decreased. So here we try to understand what molecular mechanisms enable the bears to succeed here where man fails. So our aims basically are to better understand muscle protection mechanisms in hibernating bears despite atrophic conditions, and try to translate their strategy to humans. And in a more global way, we want to better understand metabolic adaptations. So today I will show some of the results we obtained, largely using omics technologies. I will go fast for lipidomics or transcriptomics results, and I will really focus my talk on proteomics results. We are going twice a year in Sweden, and our study area is located here, approximately 400 kilometers north to Stockholm, in an area where bears are densely present. We can sample up to 5 to 6 pairs a year during summer and also during hibernation. The same bears in a given year are captured, so a bear is its own control. And we can sample blood and also take some small biopsies of fat and muscle. And all the results I will present today come from the analysis of the vastus lateralis muscle. So first, quickly, a few results using lipidomics, because today it's a question of omics. With lipidomics, we could first show how lipids are distributed among the different tissues during hibernations. And here, the shifts that were recorded were exactly the same that were known for small hibernators. So everything here seems to be common for all hibernators, be they large as the bear or small as a terrestrial squirrel, for example. We also could see that there is a depressed state of the entire eicosanoid cascade. And finally, studying the regulation of lipoprotein and cholesterol metabolisms, we could establish that despite the large lipid fluxes during hibernation, bears do not seem to develop atherogenic adverse effects. Then we also used transcriptomics. And with transcriptomics in the bear muscle, we could highlight a large number of differentially expressed genes, and notably those of the TGF-beta and BMP pathways that are well known to control the protein balance. And what we see in more detail is rather a downregulation of the TGF-beta signaling, but stimulation, or at least maintenance, of the BMP pathway signaling. And these regulations are in agreement with muscle maintenance because they would favor synthesis and inhibit degradation of proteins. What is interesting is that at the same time, we could see the exact opposite regulations in the muscle of those atrophic mouse models. And more recently, we could show, using halofuginone, that favors the BMP pathway over the TGF-beta pathway, that the treatment of the mouse was associated with reduced muscle atrophy in these mouse. We continued a bit with RNA and also studied some microRNA that pilot the fate of mRNAs, including in muscles. And I do not detail here, but the regulations we observed here were in good agreement with metabolic suppression during torpor, and also with skeletal muscle maintenance, with the activation of few protein synthesis pathway, but inhibition of certain protein degradation pathway. And now proteomics. So for proteomics, we started with this type of analysis in 2018. And at this time, there were no known sequences for brown bears, our model. The only sequences that were available were for the panda bear or for the polar bear. And so here we only relied finally to analyze our proteins on the very conserved sequence portions of the proteins. And despite that, we were able to identify 162 differentially proteins in the bare muscle, comparing winter and summer. And functional annotation enrichment analysis allowed us to show that all of these biological processes were finally altered during hibernation. If we try to organize everything here, those different biological processes, they belong to these 4 categories. So tissue remodeling, the response to oxidative stress, fuel and energy metabolism, and the regulation of protein balance. And in the following slide, I will go through these 4 main categories to show you how we went a bit further to understand what happens here. So for tissue remodeling, we started to analyze CSA of the muscle fibers, for example, and we could see that there were no significant difference here. We also could see that there was a significantly reduced contractile force of the muscle fibers, as isolated muscle fibers, but there was a reduced contraction velocity of the fibers. And in parallel, we could see a reduced ATPase activity in hibernating bare muscle isolated fibers. So to try to understand what molecular mechanism could drive these observations, we performed a single fiber proteomics sequence analysis, and this was done in 2023. Since 2019, we have sequences for brown bears now, and there had been several improvements in the assembly and annotations. So this was using the very last annotation. And here, the results could highlight the drastic remodeling of hibernating bare muscle, with notably a large reduction in the abundance of mitochondrial proteins during hibernation in the muscle fibers. And notably, we could see a reduction in the content of this myosin kinase, MILK2. And after this, we could confirm the reduction of MILK2 using Western blots, but we could also confirm that the phosphorylation of its target, RLC, was also reduced during hibernation. And this could be directly responsible of the reduced ATPase activity. Interestingly, then, we tried to analyze MILK2 levels and also the phosphorylation level of its target in a mouse model of muscle disuse, and we could find the same reduction here, and also here just a trend of the reduction of the phosphorylation of the target. So what was concluded in this study was that the energy loss in the bare muscle during winter could involve reduced myosin ATPase, a mechanism that could involve MILK2 downregulation. Then quickly, I will talk about the response to oxidative stress, which you know is linked to the promotion of muscle proteolysis, but inhibition of protein synthesis. And what we could see here from the proteomics data is a complete decrease of all, as already said, the abundance of the subunits of complex 1, 2, and 3 of the mitochondrial respiratory chains, so the main location of ROS production. But in the same time, we could see an increase in the abundance of antioxidant of proteins of the antioxidant systems, and also for chaperones and HSP. Those 3 ones are mitochondrial. So all of these results were in line with what we measured later, a reduction in oxidative damage in the muscle of bears during hibernation. And we could conclude here that limited oxidative stress promotes resistance to skeletal muscle atrophy in hibernating bears. Then we went to fuel metabolism, fuel and energy metabolism, and we first looked at the proteomics results again. And the first observation was that the protein abundance of all mitochondrial proteins here are decreased for beta oxidation, so lipid utilization, but also for near all the subunits of the mitochondrial respiratory chain. This was also true for few of the enzyme activities that we measured here. And contrary to this, and surprisingly, because this is not the case in small hibernators, for example, everything for glycolysis was rather increased. On the contrary, so this was the first striking results. But we nevertheless could propose that, as in small hibernators, glycolytic intermediates cannot feed the Krebs cycle because of this pyruvate dehydrogenase kinase, which inhibits the activity of the pyruvate dehydrogenase. So to explain and to go a bit further about these particular results, we looked at the abundance of glycogen in muscles of our bears. So here, it's the same magnification. Here, during hibernation, you may see all those black granules of glycogen, which seems to be absent during summer. And in fact, glycogen stores were increased by a factor of 3 during winter. And we could validate this observation by biochemical measurements. And the literature tells us that such a result could be due to some omega-3 fatty acids, like, for example, DHA. So we measured DHA as well as DPA. Both of them are come from their precursor EPA. And you can see that in winter bears, so in black, there is a decrease in the abundance of the precursor, but a large increase in the abundance of those omega-3 fatty acids. So here we could conclude that potentially DHA could be involved during winter in the bear muscle to protect the muscle, but the exact mechanism, of course, remains to be determined. Then I will stay on fuel and energy metabolism, but I will now focus on the mitochondrial function because, as you understood, most of our down-regulated proteins are mitochondrial. We first analyzed the function of the mitochondria using the Ouroboros instrument. And what we could first observe is we performed the analysis in permeabilized muscle fibers at 2 different temperatures, 37 and 33. So the temperature of bears during summer and the temperature of bears during winter. And as you can see, there is a coupled respiration here involving complex 1 or complex 1 and 2 of the mitochondrial respiratory chain. So coupled respiration is decreased in hibernation, whatever the temperature. So this indicates a reduced mitochondrial oxidative capacity during hibernation. And what I do not show you here is what this was less pronounced when providing the cells, the fibers with carbohydrate substrate compared to lipid substrate. So here we propose that the hibernating muscle may be adapted to lower winter temperature when carbohydrate fuels are available. Then we calculated the ratio of coupled to uncoupled respiration, and we could observe that again it was lower at the 2 temperature during hibernation, indicating a drop in the ATP-driven respiration. And here, this is the ratio of the maximal electron transfer system capacity and coupled respiration, which, as you can see, was not affected at both temperatures. And this indicates that both in summer and winter, mitochondria here in vitro operate near maximal capacity. last result here, functional result, we calculated the ratio of coupled respiration involving either both complex 1 and 2 or only complex 2. And here you can see that there was a lower level during in winter samples. And this was due to the fact that lower temperature were in fact activating complex 2, but they were also diminishing the contribution of complex 1 to respiration. So complex 2 seems to potentially be key during hibernation. And to try to explain a bit better this greater reliance upon complex 2 during hibernation, we performed proteomics, of course, to try to find some molecular mechanisms. So here, this experiment was done with the sequences from the 2022 protein database. We could highlight 300 differentially expressed muscle proteins here, almost 200 of them being mitochondrial. And as you can see here on this volcano plot, the dots in color are mitochondrial proteins, and you can see that they are below the zero. So there is a predominance of downregulation for the different mitochondrial processes. In parallel, what we did, we isolated the mitochondria of the same muscles. And we again performed a proteomic analysis. And as you can see here, the number of differential protein is drastically decreased, almost nothing. Only 8 significantly differential mitochondrial proteins were found here. So here we propose that the mitochondrial machinery is preserved during hibernation, but there is a reduced content of the total mitochondria during hibernation, what we could validate by measuring the ratio decreased here of mitochondria DNA over nuclear DNA. So only 8 differential mitochondrial proteins, but could they give us some idea of mechanisms to sustain SDH activity? First, here I show you that SDH activity is indeed maintained during hibernation, both at the total muscle level, but also in isolated mitochondria. No significance here. And from the differential mitochondrial proteins, we have different potential mechanisms to explain this. First, we could observe that despite most of the things were downregulated, there was a maintenance of the abundance of the decarboxylate carrier SLC25A10, which may help sustain this enzyme with its substrate succinate. We also could observe that that glycerol phosphate shuttle protein levels were also maintained, and this could help maintain the supply of FAGH cofactor to complex 2 again. We also observed that this enzyme FAHT1, the levels were increased in the mitochondria during hibernation, and this could favor, again, SDH activity because this enzyme is known to remove an active inhibitor of SDH. And finally, we hypothesized that knowing that lactate can enter the mitochondria without any carrier, maybe lactate could enter the mitochondria and stimulate electron transfer chain via the activation of the pyruvate dehydrogenase activations. This has already been suggested in vitro in many types of cells in 2023. This effect is without any metabolization of lactate. And indeed, we also could observe in our proteomics data that the amount of LDH were decreased in the mitochondria, despite they were increased in the total muscle. And finally, we made some new analysis to better understand what happens for protein metabolism, the regulation of the protein balance. Our first results from 2018 were pointing at strange results because we were expecting the different factors of the catabolic systems to be downregulated, but here they were rather increased in those data. So we were a bit puzzled here, and this is absolutely not apparently in agreement with what is known for protein synthesis and degradation rates during winter in bears, which are both, as you can see here, decreased. So synthesis and degradation here 1 month after denning, and here 4 months later. So we raised the question of how the ubiquitin proteasome system is regulated during hibernation in bear muscle. And we performed a new proteomic analysis of our bear muscles. And we particularly here focused on the ubiquitin proteasome system. As you can see here, we confirm the results of 2018 while being more comprehensive with the near all the subunits of the Vantes score complex being upregulated during hibernation, and it's the same for the different subunits of the regulatory particles, and measuring in vitro the maximal activity of the proteasome, we could also detect a significant increase at 32 degrees C and almost significant at 37. In parallel, for the same muscles, we isolated, we immunoprecipitated the proteasome complex, targeting a subunit of the Vantes score complex. And here, again, a proteomics analysis could show us that the results were the same with an increased, here we could see that we are around 1 here. So there is no difference in the abundance of the proteasomal subunits, both for the Vantes score complex and the regulatory particles. So this indicates us that for winter and summer samples, we got the same yield. And then considering the interactome that we will see now, there was no real bias. With all of these information from the whole muscle and from the immunoprecipitated proteins, we could make calculation to determine that there was a small but significant decrease in the amount of, in the proportion of the free 20S core complex, but small again, but significant increase in the activated forms of the Vantes of the proteasome. So here taken as a whole, and here we made calculations for every type of activated forms. So here it's close to significance, here it's significance, here it's not. We also validated here that ATP was not a limiting factor in the assembly of the proteasome complex. As you can see here, we could measure increased levels of ATP during winter in the muscle. And here, we also looked at one of the more potent inhibitor of the proteasome assembly. And here, you can see that the abundance of this inhibitor is decreased, but at the whole muscle level and also among immunoprecipitated proteins. We then, after the proteasome, got interested in ubiquitination. You know that to be degraded by the proteasome, the proteins are to be targeted using a ubiquitination process. As you can see here, there was no real difference in protein ubiquitination process here for total protein ubiquitination, or here for K48 ubiquitination, which is one of the specific type of ubiquitination that target proteins to the proteasome. And finally here in our proteomics data, you can see how ubiquitinating enzymes vary in abundance. So there are some differences during winter compared to summer. We cannot know if these changes provide some specificity. We cannot know if some of them could be K enzymes, because in some situations, only one downregulation or only one upregulation here is sufficient to either protect or degrade the muscle. Ubiquitination, but also deubiquitination is involved in the process in the ubiquitin proteasome system. And there are two types of deubiquitinating enzymes, those that favors protein degradation by the proteasome. And here you can see an increase, a large increase, significant increase for this one, but no change for this one. And the other type of deubiquitinating enzymes are enzymes that are favoring substrate deubiquitination. So proteins, ubiquitinating proteins in this case, escape degradation by the proteasome. And here we could see few changes only with some upregulations here. So maybe those enzymes could maybe be involved in counteracting protein degradation, at least maybe for some specific proteins. Here we don't really know, of course. And finally, if we look a bit more in detail in the interactome as a whole, the interactome of the proteasome complex, you can see again here in this heat map that most of the proteins, they are decreased in abundance. So it seems that there are less proteins associated with the proteasome during winter. And those downregulated proteins, they notably especially belong to the protein synthesis system, to the mitochondria again, and also to some proteins involved in muscle contraction. The few upregulated proteins here belong also to the protein synthesis rate, and for some others are known to contribute to DNA organization with numerous histone, for example. And on this detailed view, I just would like to highlight the black proteins, the black dots here that are all downregulated. And these are the major sarcomeric proteins. So it seems like during winter in bear muscle, the sarcomeric proteins interact less with the proteasome. So potentially they could be protected. And ubiquitin proteasome probably remains in an active form to promote potentially quality control of the newly synthesized proteins, potentially in anticipation to emergence. So in the coming months, we will get new results from proteomics that I cannot show you already today, because we are analyzing a data set of phosphoproteomics that we performed on bear muscles, in connection with some of our previous works showing that human muscle cells cultivated in vitro and incubated with hibernating bear serum, so either winter bear serum or summer bear serum, they exhibit, for the cells incubated in hibernating bear serum, an increase in size when we talk about skeletal muscle cells. And if we talk about cardiomyocytes from mouse this time, when incubated in the bear serum, they were protected against ischemia reperfusion stress. So here we, of course, would like to identify the compounds in the blood that could provide such effects. And for this, we performed a complete proteomics and metabolomics analysis of the bear serum during winter and summer. And so here we have more than 600 differentially abundant proteins, because we used a more refined proteomics analysis strategy. And we have a total of 150 differentially abundant metabolites. So we are in the process of going a bit more in detail in those results. And in parallel, we treat different types of cells with our bear serum and measure various different effects So potentially these effects are analyzed functionally, but also in parallel with some omics analysis. So to conclude today, I tried to show you that in this physiopathological situation of hibernation, there is metabolic adaptation and skeletal maintenance in hibernating bears, which appears to involve a multifactorial regulatory program. And you could see that a large part of our results derives from proteomics data, which provide many complementary results, which support most of the time, if not always, the functional findings that we can obtain. Of course, the combination of proteomics with other omics is expected in the future to give us a more comprehensive picture of what happens in the muscle during winter. And we hope that our results could stimulate some new solution to help prevent or fight human muscle atrophy in the in various people among the population, so obese or emaciated, sedentary people, people immobilized in hospital beds, and so on, aged people, and astronauts. So finally, I take this opportunity to thank here all my collaborators on this project, my sponsors, and of course you for your attention, and I will be happy to take questions.
Thank you, Fabrice. Maybe I can just ask about the myokines, which may impact on the muscle and also other organs. So have you already started to look to these myokines?
We want to, but it's difficult to find tools to really assay molecules that are often found in low abundance in the plasma or serum. So together with the analysis of the serum, I mentioned proteomics and metabolomics, but we also tried to validate a series of different ELISA to assay different hormones, so among which potentially myokines, yes. But this is still in the process of the examination of the result.
I have a question because you show that the instance can change. I saw that the H1 is increased.
It's increased in the interactome of the proteasome. So it's like the different histones could be more degraded.
Degraded.
Yes, during hibernation.
But have you thought about analyzing with using ChIP-seq which loci?
Yes, we would love to. It's still not done. What we did already is just analyze the methylation proportion of some histones, notably H3, for example, which seems to be increased during winter.
Okay.
But we are at the very beginning of all what we want to do around epigenetics and.
Thank you.
Amazing study. What do you expect in anatomic different muscles? Would you expect that there are differences among muscles? And for example, what about diaphragm? Because they still breathe. Less frequent, I would say.
Apparently, morphologically, there's not so much that happens. You could see an electron micrograph picture. There's not many things that happen. For example, the fiber types, they do not change between winter and summer. It seems that at the sarcomeric level, there is not so much that happens. So for sure, this is something to look at more deeply in the next years. But for now, we don't have many differences that are really visible. What I could see, I measured since few years. I've also been measuring the muscle tone, for example, and it seems like the muscle tone is at higher level in winter.
So I have a rather stupid question. So what is the trigger for hibernation? Is it temperature or is it daytime periods? Because there must be a metabolic trigger.
It's not really known. What is observed is that the different features of hibernation, it's not a switch like this. Everything starts to decrease for some, for example, for heart rate, it already starts to decrease 1 month before denning. So, and the entry in the den is really finally linked to the amount of snow. This means when the bear cannot find any food any longer. So, but everything is is very progressive. What we can read usually is temperature, the decrease, of course, in food availability, and the photo period that is changing. But that's it. We don't have many triggers, and it's a bit the same for den exit, because everything is also growing up slowly but progressively, at least 3 weeks before emergence for all the parameters. It seems like that den exit has been related with kind of a temperature incomfort, because this was related with the increase in the external temperatures. But molecularly, physiologically, we don't have real information there.
What happened to the brain?
In bears, we don't know anything.
Really nothing?
Really nothing. I tried to
need a bear neurologist.
I tried to stimulate some recordings of the brain activity, but it's super hard to find. We cannot do this in the wild, so this has to be done in a zoo or in a park, and I did not find the park. I searched in all over Europe. It's impossible to find a park willing to do this. Apparently, we have colleagues from the US that are doing kind of parallel work with black bears. They have been trying to do this in black bears since 2011 or 12, something like this. It has been apparently very challenging for them. I heard that they may have now some results publishable, so maybe we will know more in the coming weeks. I don't know.
One final, the hibernation period, is this individualized? So it's just one bear has its absolutely individualized hibernation period?
Yes.
Okay, yeah, thank you. As you were talking about black bears, many of you know that polar bears don't hibernate. And due to climate change and things like that, lately there are more and more hybrids of polar bears and grizzly bears. Do you know anything about their hibernation and what happens in their
Polar, for the hybrids, I don't know. But for the polar bear, you're right. They do not hibernate except the mother when she's pregnant.
Then you would
because bears give birth during hibernation, and they have a very rich milk, and this is the better solution to save energy, because when they get out of the den, they don't have energy anymore to feed the young. So,
and I have another question. I mean, you cannot directly compare hibernation of rodents and carnivores, say, and of course there are some rodents that do have a very extreme hibernation. I think of dormouse, edible dormouse, very famous. But wouldn't it be more cost-effective and less dangerous to study dormice instead of bears?
So for the dangerous part, for sure. But for the rest, I'm not sure. So there are some differences. You're right in between large and small hibernators. For example, the bear body temperature is decreasing, but not that much. the temperature of a small rodent hibernator can decrease to the temperature of the ambient temperature, so close to zero often. Small hibernators, when they hibernate, when they are in torpor, you can take them in your hand, they will not awake. Bears remain sensitive to the environmental stimuli. And here we could show that in fact, the endocannabinoid system is regulated in a different way, which could contribute to this point. And the very last difference is that most of, not all, but most of the small hibernators, they rewarm and they have periodic euthermic bouts during the hibernation, not the bear. The bear remains in this torpor state during those 7 months. So now, yes, it could be more dangerous, but regarding the amount of sample you can collect, you would need a very, very large amount of animals in your animal facility compared to here, the collaboration with a consortium who is following and capturing the bears from this population since 40 years. So
anything about the orexin system? So in Sierra, you can.
I don't know anything in hibernators, but maybe, but I don't know this literature.
Okay. Thanks a lot then, Fabrice.
Thank you.
Thank you. So we'll have a small break and then we come back at 11.
I just have a communication. So do not forget to sign the paper for the attendance list. So meaning that Joanna will provide us the sheet to be signed, and you have to be signed for yesterday if you were in, yesterday evening if you were in, and for people that are joined today, just for today. Thank you.
We'll discuss about inflammatory myopathies. So thank you, Alain.
Thank you very much for the invitation and the introduction. So yes, today I would like to provide you first an overview of what is inflammatory myopathies. And I would like to focus on 2 studies that we have the chance to perform with collaborators to try to unravel the mechanism of this disease. And in these 2 studies, we use Omics data to achieve our goal. So first of all, what is inflammatory myopathies? Actually, it's some diseases that are quite young in the history of the medicine, because you see here, so the example is on purpose, of course. It's a doctor from Strasbourg. His name is Paul Epp, and he was among the first to introduce this concept of polymyositis. So this is a synonym of inflammatory myopathies to describe the patient who has muscle quickness and inflammation of the skeletal muscle, and the origin at this time was unknown. And actually, you will see that we have some work to perform today to understand this inflammation in the muscle. But today we have diagnosis criteria, and basically inflammatory myopathy, this is first a myopathy, so it's a weakness of the muscle, of course, and dysphagia, and increase of muscle enzyme. So this is creatine kinase, and as you know, this is an enzyme that is located in the muscle fibers. And when the muscle fibers are broken, you can measure an elevation of this enzyme in the blood of the patient. So this is non-specific. Actually, this is what we see in numerous myopathy. But what is particular in this disease is that this is an autoimmune disease. And currently, the criteria that we have to to say that the origin is autoimmune is that there is extramuscular involvement, and in the criteria, this is a peculiar erythema of the skin. The other criteria is the presence of autoantibodies. Only one is included in the current diagnostic criteria, but actually today we have discovered about 30 different autoantibodies in this disease. And finally, this is peculiar finding in the muscle biopsy. So this is peculiar to this disease. To diagnose the disease, we usually perform a muscle biopsy. So this is helpful to perform the diagnosis of the patient of the disease, but it is also useful to perform research and to understand the disease of the patient. These diseases are severe, and you see here the mortality versus the general population. Globally, it is increased by 4-fold, and in the first year of the diagnosis, the patient have a 10-fold increase to die as compared to the general population. So this is severe. And what are the causes of the disease? Actually, I have no time to go into the details, but in numerous cases, there is an associated cancer in a paraneoplastic link. Cardiovascular events are frequent, and this patient can develop inflammation of the lung. And this is the 3 causes of mortality in this patient. And we do our best to able the patient to survive, and even in the patient who survive, there is a decrease of quality of life versus the general population. And here it is the muscle, the joint, and the skin involvement that are predictor of a decreased quality of life. And so this is severe disease, but these diseases are treatment orphan. Actually, in France, there is only one approved treatment that is intravenous immunoglobulins. And this is not approved for all the patients. It's approved for a peculiar subgroup of patients that have, we will talk about this later, a subgroup that is called dermatomyositis. And this is for severe dermatomyositis. So most of the patients are like treatment orphan. And we have review with the ERN dedicated to autoimmune disease, including inflammatory myopathies, the current clinical practice guideline. And what we show here is that the current treatment are actually empirical. So not surprisingly, they are partially effective and they expose to numerous side effects. So in this context, the aim of our research and our colleagues that are involved in this field is really to understand what are the mechanisms that underlie this disease, to propose the patient treatment that target pathophysiology of the disease and not to use empirical treatment that are frequently repurposed from other autoimmune disease to the inflammatory myopathy. And so we develop 3 approaches. The first one is epidemiological, second is clinical, and the last approach that we developed for the disease is translational. I will go quite fast on the 2 first aspects, but I just would like to present you this research as a further introduction to the disease. Because it is important to know that on the epidemiological point of view, inflammatory myopathies are rare diseases. You see here a meta-analysis of the incidence that is reported in the literature. And there is actually less than 10 persons per million inhabitants per year that are diagnosed with this disease. So it's a challenge to study this disease. And we performed a study in Alsace to determine whether the incidence in our region is the same that in the rest of the world. And the answer is yes. We record 8 million new cases per year in Alsace. And what we see is that the current diagnosis criteria actually underestimate a bit the incidence by 25%. So yes, I show you these two points that a challenge in the research of this disease is the rarity of the disease. And from the clinical point of view, we and others have performed a lot of description of the disease that currently indicates that the first description in the 19th century of one disease with inflammatory lesion of the muscle is actually incorrect, because these diseases are a group of heterogeneous disease. Heterogeneous disease based on the clinics, because the importance of the myopathy and the importance of the extramuscular involvement varied amongst patients. And as I have mentioned, there is a lot of autoantibodies, and these autoantibodies aggregate with the clinics. So it means that when the patient have a peculiar phenotypes, he generally have a peculiar autoantibodies. And the third layer of this classification is that the histopathological lesions are also different. So this classification is a schematic view of the disease, but you have to know that actually today there is no consensual classification of the disease. And this is another challenge of the research in inflammatory myopathies, that is that disease is actually heterogeneous. But still, we have to understand the disease. So we perform translational research and trying to overcome this challenge. And the first one I would like to focus on has been performed in this context. We and others have shown that in the muscle of the patient, so that we perform for the diagnosis, we found inflammatory infiltrate with immune cell, and we found also muscle lesion. And in the fibers, what we see is that the patient have mitochondrial dysfunction. And this mitochondrial dysfunction is something that decreases the exercise capacity of the patient. So this was not a surprise. But we also have data that indicate that actually mitochondrial dysfunction can contribute to the inflammation of the muscle. But still, it was not so clear whether the immune cell cause the mitochondrial dysfunction, or is it the other way? Is it the mitochondrial dysfunction that participate to the immune cell inflammatory infiltrate? And so to approach this question together with the team of Olivier Boyer in Rouen, we studied a mice model, a spontaneous mice model of inflammatory myopathies. So this is a node mice with a knockout of ICOS, so ICOS null node mice. And these mice developed something that resemble inflammatory myopathy that we see in human. You see here that at 35 weeks, there is an inflammatory infiltrate in the muscle. And these mice developed weakness of the muscle. And in the muscle, we can measure at 35 weeks, an increase of interferon beta and interferon gamma cytokine in the muscle. And this is something that we also see in human. And so to understand why these mice spontaneously developed weakness, inflammatory infiltrate, and increase of interferons in the muscle, we perform a muscle proteome of these mice. And what we see is that oxidative phosphorylation deregulation was the top one deregulated pathway of the muscle of ICOS null node mice versus the control. And I have not the time to go into the details, but further experiments confirm that there was mitochondrial dysfunction in the muscle of these mice. And this was based on histochemical staining of the mitochondria, a functional recording of mitochondrial respiration, as our colleagues show previously with Ober's measurement. And finally, with electronic microscopy, we were also able to highlight mitochondrial dysfunction of a mitochondrial morphology abnormality in these mice. And what I would like to point is that very interestingly, this mitochondrial dysfunction was an early event in these mice, because you see that it was detectable at week 8. So this actually precedes the inflammation. And this indicates that mitochondrial dysfunction is an earliest event as compared to inflammatory infiltrate and to interferon beta and gamma in the muscle. And so to further study the link between this mitochondrial dysfunction and the inflammation, we perform spatial transcriptomic analysis of the muscle of the patient. And we compare fibers that were in proximity, but not directly adjacent to the immune cell cluster, versus some myofibers that were directly adjacent to immune cell cluster. And the comparison of these two areas of the muscle show that there was an increase of mitochondrial dysfunction in the fibers that were directly adjacent to the immune cell cluster versus the other fibers. So this shows that actually this mitochondrial dysfunction prevails in the myofibers adjacent to the inflammatory cells. So there is an association between these inflammatory infiltrates and the mitochondrial dysfunction. But what is the causal link between the two? So to study this, these ICOS null mice were treated with N-acetylcysteine, and as you know, this is a ROS buffer. And what we see in these mice is that N-acetylcysteine, as we expected, was effective to buffer the ROS. You see here in blue the group that is treated with N-acetylcysteine. And this was associated with lowest mitochondrial dysfunction on the histochemical staining of COX. This was also associated with a decrease of the inflammatory infiltrate in the muscle. This was associated with a decrease of the level of interferon gamma in the muscle. And finally, this was associated with an improvement of the phenotypes of the mice. The treated mice were less weak than the untreated mice. So this experience show us that in this model, the inflammatory infiltrate is dependent and the weakness is dependent on the ROS. But on the other hand, we also treated human myotubes exposed to interferon gamma. So this is like an in vitro model of the myopathy. And we treated these myotubes with JAK inhibitor. And as you know, the JAK is the proteins that are directly downstream the receptor of interferon gamma receptor. So actually what we do is to block the interferon gamma pathway in this in vitro model. And the mitochondrial abnormality on electronic microscopy that were induced by interferon gamma were actually blocked with two JAK inhibitors, that is ruxolitinib and baricitinib. So these additional experiments show that interferon gamma is actually inducing the mitochondrial dysfunction. So what this experience show us in a nutshell is that myofibers, mitochondrial dysfunction, and interferon gamma are actually interconnected in a self-maintenance loop, at least in this mice model of the disease. So this is an example of what we have learned from omics in a mice model of the disease, but obviously mice model is not human disease. And so we also studied samples from patients. And for now, we have focused on a subgroup of the disease that is called dermatomyositis. And dermatomyositis, as compared to the other group of inflammatory myopathies, have a peculiar histopathological pattern. This consists of lesions that selectively affect the muscle fibers that are located at the periphery of the fascicle. And these perifascicular fibers are characterized by different modifications. The first one is an atrophy of these fibers as compared to the rest of the fibers. They express human leukocyte antigen type 1, and this is not normally expressed by muscle fibers, by mature muscle fibers. And they also have mitochondrial dysfunction. And finally, they express interferon-stimulated genes. But the origin of these 4 modifications that are the hallmark of the dermatomyositis was unknown. And so to unravel the mechanism that lead to this modification, we performed here in IGBMC, thanks to the platform that was developed by colleagues, so thank you. Experiments in which we perform microdissection of these fibers who present the lesion of the dermatomyositis, and of fibers that are spared by the disease. And we perform this in a group of patients with dermatomyositis and 2 control groups, patients with other inflammatory myopathies and patients without neuromuscular disease. And the transcriptome of these fibers was performed, and bioinformatic analyses were performed. And what we show first is that in patients with dermatomyositis, there was an increase of 18 genes, and 10 genes were decreased. And as you see here, this is very specific of the disease, because in other inflammatory myopathies, there is hardly no difference between the fibers that are located in the periphery of the fascicle and the fibers that are located in the center of the fascicle. And what was a bit surprising, this is not a topic of the talk, but actually what we see is that in patients without neuromuscular disease, there is also a peculiar signature, transcriptomic signature of the perifascicular fibers. But this is completely lost in the patient with non-dermatomyositis, myositis, and in patients with dermatomyositis. And when we look at into the pathway that were the most deregulated, we found autophagia, mitochondria, and proteasome. And I don't know if this talk, if it was on purpose that I just talked after the hibernation of the bear, but actually it's funny to see that we found some similar deregulated pathway in dermatomyositis. and in the bear that during hibernation. And we because we are doctor, and because there is no label drug for this patient, we always think whether we can translate these findings to improve the treatment of the patient. And so to this aim, we perform a drug repurposing analysis and asking to 3 different databases if there is already approved drug that might be able to reverse this transcriptomic, this pathological transcriptomic signature. And we found 9 drugs that were predicted by at least 2 data sets as drugs that might reverse this transcriptomic signature. And what were these drugs? The first one was prednisolone. So this is not a breakthrough in the treatment of the myositis. But I think we think that it is a validation of our data because, as you may know, prednisolone is a standard of care of this disease. So we have found the standard of care with using this unbiased analysis of the matter. So this was quite good. We also found a JAK inhibitor. And so again, this was not new, but I think it's a validation of the analysis because there is some several case report and small series that reported efficacy of JAK inhibition in patients with dermatomyositis, and there is currently ongoing trial. But what was new is that we found 2 proteasome inhibitor predicted as therapeutic to reverse the transcriptomic signature that we observe in our patients. So this is in silico prediction. Of course, it should be validated and first at the preclinical level. And to this aim, we have set up an in vitro model of the disease. This is myotubes that are treated with interferon beta. And actually, when we treat myotube with interferon beta, we reproduce the dermatomyositis lesion that we find in the perifascicular fibers. That is, of course, interferon stimulated genes because we treat with interferon beta, but also atrophy, HLA1 expression, and mitochondrial dysfunction. And these myotubes were treated with ixazomib or vehicles, and we assess the ability of ixazomib to reverse these dermatomyositis-like lesions that we see in vitro. And the atrophy was reversed by ixazomib treatment. The expression of MHC1 was abolished by ixazomib. And the mitochondrial dysfunction recorded here by the oxygraphic analysis of the myotubes were also reversed by ixazomib. And there was no statistically difference in the interferon stimulated genes. So what we see here is that in this in vitro model of dermatomyositis, ixazomib reversed the interferon beta-induced dermatomyositis-like lesion, and this was independent of the interferon stimulated genes. And finally, we use an in vivo model to assess whether ixazomib is also effective in mice. And this model of dermatomyositis is based on the immunization against protein C. This is a protein of the muscle. And these mice developed at day 40 weakness, increased CK, atrophy, CMH1 expression of the muscle fibers, inflammatory infiltrate, and mitochondrial dysfunction. So these are all characteristics of dermatomyositis. So this is why we use this mice. And this mice was treated or not with ixazomib, starting at the peak of the disease, so a therapeutic design. And what we observed is that ixazomib was able to reverse the muscle quickness in this model. CK level was normalized with ixazomib. And a parameter that approach the atrophy was also reversed by ixazomib. And the inflammatory infiltrate in the muscle was abolished or at least importantly decreased in the muscle of the treated animal. So this shows that proteasome inhibition is effective in an in vivo preclinical model of dermatomyositis. And so to summarize what show these two experiments that were firstly based on omics is that in dermatomyositis, we can inhibit, of course, the pathways that are downstream of the interferon receptor. We can buffer ROS, and we can also inhibit proteasome to treat the disease, at least in a preclinical stage. And so we now have a project to go further into the details of the omics, and we include patients that are untreated with inflammatory myopathies, and we also include patients that have no neuromuscular disorder, and we record standardized clinical data for this patient. We take PBMC serum. We also collect the muscle biopsy, and we follow this patient during a 3 years follow-up to record the outcome of the patient. And then at 3 years, we record once again the clinical data, and we sample PBMC serum. We do not sample another muscle biopsy because from an ethical point of view, I think it's not acceptable. And so currently we have 62 inflammatory myopathies patients that are included in this project. And we have the idea that applying omics technique in this sample with the use of the standardized clinical data recording on a prospective manner will help to refine the classification, because currently the classification is based on clinic, autoantibodies, and histology. But if we go into the molecular signature of this patient, we may find a way to improve the classification of the patient. It is also important to identify a predictive biomarker in this disease, because the outcome and the trajectories of the patient are heterogeneous, with some patients with mild disease that are easy to treat, and others that are very refractory and will eventually die of the disease. And so we need a biomarker, baseline biomarker, to predict this outcome in order to tailor the treatment of the disease, starting at the first prescription of the drug for our patient. And we have the hope that this approach will further reveal pathways that are underlying the pathology of the disease, and to reveal candidate drugs that may improve the patient. And so with this, I would like to conclude. I hope that I was able to illustrate how the use of omics approach can be useful tool to unravel the mechanism of inflammatory myopathies that are rare and treatment orphan disease. And I have not the time to develop it, but if you want, we can discuss it. I think that there is new avenues in these omics techniques, and what we try to do now is to combine the information gained from several samples, as I show you, PBMC plus muscles that are sampled at the same time in the same patient. And there is some challenge that are present in this disease. I have presented some, but one I have not the time to develop is that there is some subgroup, at least one subgroup, in which when we perform the transcriptomic analysis of the muscle, we do not have a transcriptomic signature. And so we have some data that indicates that actually the modification in the muscle disease is derived on the enzymatic layer, but not on the transcriptomic layer. And so we try to develop a metabolomic approach for this peculiar subgroup. And last but not least, I would like to thank all the colleagues who participate in the research with our team. All this data, I would not be able to present all this data without the help. So here I would like to thank you, to thank them all. And I will be happy to take some questions if you have.
Thank you, Alain. Is there any questions?
Yes. Thank you very much for your presentation. I am curious about the trial because as you highlighted, it is very important to classify the patients, otherwise it is difficult to understand. But what about outcomes, things that biomarkers, because this of course is very important, but it is also very challenging. Can you tell us more?
Yes, yes. So thank you for the question, because you point, I think, a crucial point for the trial in this disease. Currently, there is a lot of trial for this disease. It was not the case 3 years ago, but now we have more than 10 trials in a very rare disease, so it's quite competitive. And one of the challenge is actually, as you pointed to classify the patient. And in my opinion, we have to improve the classification in this trial, because definitely we have refined a bit the subgroup of the patient, and especially we have split the disease very schematically on a group of the disease that is dependent of the interferon pathway and a group that is independent of the interferon pathway. But definitely we have to refine this classification. There is an ongoing work with the ACR, so the American College of Rheumatology, and the EULAR, that is a European group, to refine this. But in my opinion, the omic approach will be very helpful to this aim. So this was for the classification. But just to finish on this, I think it's a very important question for the patient, because there is also on the regulatory aspect, if you have a diagnosis as a patient, will you have access to the drug that is labeled with another wording? And here there is some heterogeneity even in the wording of the disease. And there is another ongoing work to harmonize the name of the disease in order to provide the access to the future drug that will be I hope approved. And the second is regarding the outcome. So this is a bit technical, but basically currently what we have is a consensual tool to assess the response to the treatment in human. And it is based on, of course, the weakness, the level of TK, but also the activity on the extramuscular involvement. So how much important is the edema, how much inflammatory infiltrate in the lung on CT scan is important, and how much the joints are painful and swollen. So this is good because with this outcome, we were able to perform positive clinical trial. The example is intravenous immunoglobulin. But on the other side, what we have to refine is to personalize these tools to the different subgroup of the patient, because as I presented, there is some patients that are extremely weak. So in this patient, it is all about weakness. And others are not weak, and they have interstitial disease. So they do not they are not concerned by the weakness. And at this moment, we use the same tool to measure different reality for the patient. So yes, this is my answer. I don't know if that.
No,
if
no, I think it is because this is something very important. The stratification of the patient is going to interfere with your analysis, the analysis of the result. Because if you group the patient in a different way that it is not correct, you are not going to understand. Thank you.
Very interesting presentation. I was wondering, you can classify the different inflammatory myopathies with autoantibodies. This means that there's some type of targeting for these proteins. For example, for the inclusive body myositis, you have CN5A1, something like this. Do you know if these proteins in the muscle, they are dysregulated somehow?
Yes. Yeah. So thank you for this question, because this is a very hot topic, and that I have not time to present it, but I can go into some insight. So here's the classification, and here. And yes, so here there is a lot of target, and the majority of these targets are nuclear autoantigen or cytoplasmic antigen that are involved in the process of the transcription. And for example, all this antisynthetase is autoantibodies that target amino acid RNA synthetase. And G1 is again histidine, PL12 is again tronil, and so on. So it is quite challenging to understand how an autoantibody is targeting. So first challenge is whether or not the autoantibodies target is ligand in the cell. Is it possible to enter the cell and target the ligand? So this was not proven. And the other is, if it is the case, what is the link between the amino acid RNA pathway and muscle weakness? And in other disease, especially autoantibody immune necrotizing myopathies, we have a target that is HMGCR. And here we have, so it's an ongoing work, but I show you because the paper is in revision now. As you know, so this is the subgroup. You have antibodies targeting HMGCR. And as you know, HMGCR is the cholesterol pathway. And the link between the two is not known. But we have a frequent myopathy that is statin myopathy, in which the statin block HMGCR. And we have necrosis of the fiber, so quite similar disease. And recently, it was shown that when you have a mutation that disrupts the activity of HMGCR, you also have necrotizing of the fiber. So here there is a quite obvious hypothesis that is actually the autoantibodies enter the cells and they disrupt HMGCR and lead to the myopathy. And so briefly, yes, we have generate autoantibodies against HMGCR in the rabbit, and we show that actually in vitro at least, the autoantibodies decrease HMGCR activity. And we perform the same experiments with antibodies that we gain from the patient during plasmapheresis. And here again, we have a decrease of the disease. And we treated myotubes with pravastatin, these autoantibodies, and we have accumulation of lipid droplets. So this resembles statin myopathy. And the results are not on the slide, but we have also demonstrated necrosis of the muscle fibers when we increase the dose and the time of of exposition. And finally, in the patient, we have seen that this lipid accumulation in the fibers is a hallmark of this myopathy of this subgroup. And again, this was some statin myopathy. So this indicates that, yes, the autoantibodies in this subgroup is directly pathogenous. And this is the subgroup on which I was talking about. When we perform transcriptomic, we see nothing, because actually it's like all events are located on the enzymatic level. And we have now the project to perform metabolomic to understand what's going on in the muscle.
Thank you for an interesting presentation. My question is, are there any pattern in the immune cell subtype which you observed through your omics analysis, which could be a little helpful to identify the biomarkers? Has there been any immune cell, like B cell, T cell analysis on that aspect?
Yes. So on the transcriptomic level for now, I have no answer. And looking in the literature, it's there is conflicting data whether it is more a B cell, T cell in the different subgroups. So we have a project to decipher this. And what we would like to do is in the prospective cohort, we have sampled PBMC, and we would like to perform aurora analysis, so deep phenotyping of the lymphocytes, to highlight whether there is some peculiar deregulation in the population of the lymphocyte, at least in the blood. And then I think we will be able to go in the muscle and to see whether this population are also present in the muscle. So this is an ongoing work. What I have to say is that there is currently several drugs that targeting B cell population that seem to be effective. The first one is rituximab. It's a drug that targets CD20 and destroys the CD20 positive B lymphocyte. And there is also data showing that the lymphocyte expressing CD19 are also important, because there is case series in which when we targeted these lymphocytes, the patient improve. And as you may be known, there is some, so this is very preliminary data, but several teams have reported the efficacy of CAR T cell in this disease in very refractory patients. So two CAR T cell have shown, I've shown, this is not a demonstration, this is case report, so we will see. The trial is going on. But at least there was very refractory patients who were improved using CAR T cell targeting CD19. So this is plasmablast and BCML. So this is plasmocytes.
And just for the drug repurposing analysis, has there been any overlap studies between this IIMs with other autoimmune diseases where inflammation could be the effect of those autoimmune diseases in order to just for the drug repurposing?
May you please repeat the question?
Has there been any comparative analysis between the IIMs and other autoimmune diseases in which inflammation is the effect of those autoimmune diseases?
And you mean comparison of the muscle or the PBMC or the serum or?
The muscles?
Yeah. Yes, so I love to have these samples, but actually on the ethical point of view, we cannot sample the muscle of a patient with autoimmune disease that do not translate into a myopathy. This will not be ethical. But on the experimental point of view, yes, I think this will be very informative. But yes, this is patient, so I do not have the answer. But what I can tell you is when we compare the PBMC, this importance of interferon pathway is also seen in other autoimmune diseases that resemble a bit inflammatory myopathies or at least dermatomyositis. It is lupus, systemic lupus. But it is not exactly the same type of interferon. So it resembles, but still there is some difference. And that's why I think we do not yes, we are happy to take drugs from lupus and repurpose for dermatomyositis, but we should do better, and we should go into the mechanisms that are proper to the disease to have a dedicated drug.
So I'd like to challenge you on one thing. So what is the link between, there's another good story built up currently about the complement system in dermatomyositis. So what is the link to your interferon gamma signature you've seen? Do you have any ideas?
Yes. So the complement was studied a lot in inflammatory myopathies with two aspects. In some subgroups, there is deposit of complement on the capillaries. And there was an hypothesis that actually this is a complement-driven disease that lead to destruction of the capillaries and then ischemia and then atrophy of the perivascular fibers. But there was another hypothesis that is when you have ischemic reperfusion events in the muscle, it actually leads to complement deposit. So actually the disease can be the sources of the complement deposit, and the complement deposit is not the primary driver of the disease. And there was two trial testing different inhibitors of complement in dermatomyositis, and the trial were negative. So in dermatomyositis, it seems that complement is not primary driver. And the other subgroup in which complement is involved is necrotizing myopathy. So the disease I just showed you with anti-HMGCR. Some also have described a deposit of complement on the fibers. And because of the autoantibodies, they say, yes, actually, the autoantibodies do not enter the cells. They're located at the sarcolemma, and then they activate the complement by the classical cascade. And yes, it was very inspiring, but and they perform a trial with zilucoplan, so a complement inhibitor that is approved from myasthenia gravis. And here again, the trial was negative. So it seems that in this disease, the complement is not a primary driver, but it is maybe rather a consequence of the other events.
And one final question. Did you find titin coming up? Because that was in former proteomics, one of the mark of the perivascular atrophy.
Thank you for the question. Yes, we look at this protein because in dermatomyositis, there was a work, an American work, they have performed microdissection and proteomic study of the perivascular fibers, and they found that titin is actually decreased in the perivascular fibers, and we found the same result at the transcriptomic level. But yes, when we analyzed all the pathway, it was not on the top, but we reproduce this result, yes.
Thanks a lot, everyone. So thank you for the speakers. So we close the workshop on the omics and muscle, and I thank you all the participants of this workshop.